Hello, my name is Stefan Stark. I'm a PhD student in the Biomedical Informatics Lab at ETH Zurich, and today I'll be presenting SCIM, Universal Single Cell Matching with Unpaired Feature Sets. This is a framework designed to integrate multi-omic single-cell datasets. Single-cell technologies are a recently developed class of methods that are able to profile samples at the cellular level. Consider a heterogeneous sample of cells. This sample contains cells that exist in several different states, which could correspond to, for example, cell types, here represented using colors. If this sample would be profiled using traditional bulk methods, then a single measurement would be taken as a joint signal across all cells. But this sample is composed of cells across a distribution of states, which this measurement cannot capture explicitly. Single-cell technologies, however, profile cells individually and thus can explicitly capture such signals. In this cartoon, the single-cell technology reveals that each cell type carries its own signature in feature space. These signatures are represented using the colored histograms. Data generated by these technologies can often be represented as a matrix of cells by features, where each row corresponds to a single cell in the original sample. These technologies have advanced fields by providing data better suited to study complicated phenomena, such as cellular development and tumor heterogeneity. Currently, many different technologies exist and are able to measure cell states on the genomic, transcriptomic, and proteomic levels. Additionally, imaging technologies can measure cells on a spatial level. Measurements from these technologies represent unique but incomplete perspectives on the complicated cell state. Integration of these perspectives would provide a holistic view of the cell and its environment, allowing for analysis that would not have been previously possible. For example, consider profiling cells using both single-cell RNA-seq and IMC technologies. Single-cell RNA-seq measures the gene expression within a cell across thousands of genes. This provides information on complex underlying mechanisms such as pathway activity. Meanwhile, IMC technologies image cells stained by a handful of protein markers. This technology captures spatial relationships. However, the small number of markers makes it not possible to compute something like pathway activity. But by integrating these two data sets, relationships between the pathway activity informed by the RNA-seq technology and spatial effects informed by the IMC technology can be studied. However, integration is in general challenging, since most technologies consume their sample and cell correspondences between the datasets is lost. To integrate such datasets, we introduced the SCIM framework. SCIM stands for Single Cell Integration via Matching. It assumes that a sample is distributed uniformly to two or more technologies. These technologies consume their inputs to generate a dataset that can be represented as a cell-by-feature matrix. Datasets are integrated by identifying cell pairs across technology. Here they're represented using shapes. Crucially, SCIM does not require that technologies profile across the same set of features. For example, one technology might measure gene expression, while another might measure protein levels. SCIM scales to the large datasets these technologies can generate, often on the order of hundreds of thousands of cells, and it's able to integrate two or more technologies. Since each technology receives a similar distribution of cells, SCIM assumes that the underlying structure, here represented using the color clouds, is shared between all datasets. This assumption is key, and it's what allows SCIM to integrate datasets without requiring shared features. Integration proceeds in two steps. In a first step, the shared underlying structure is reconstructed and aligned within a low-dimensional latent space. This latent space is invariant to the technology. This means that similar cells profiled by different technologies are placed in close proximity, while the distribution in this space reflects the underlying structure of the sample. To construct this space, we use a variational autoencoder framework with an adversarial component in the loss function to encourage the alignment of each technology's distribution. Encoder and decoder networks are assigned to each technology, and their architectures are tailored to their respective data types. These networks operate as they would in a variational autoencoder. The encoder maps input data into the low-dimensional latent space. An embedding is sampled from the encoder, and the decoder tries to reconstruct the input from this embedding. These networks are trained together to minimize the reconstruction error. Meanwhile, a single discriminator, phi, is trained to identify the technology of embeddings. That is, phi discriminates between the embeddings of one technology from the embeddings of other technologies. Technology invariance is imposed by training the encoder to also fool the discriminator. When trained successfully, SCIM produces embeddings that are well mixed and reflect the underlying structure of the original sample. Finally, SCIM scales to large datasets and feature spaces by gradient-based optimization schemes. SCIM is an adversarial model, and training such models can be difficult. To help train SCIM, we address two major challenges to optimization. The first challenge we address is correctly aligning substructure in the data. Consider, for example, that we wish to align a single technology to the substructure defined by these colors. SCIM could produce embeddings represented by the gray hexagons. These are colorless since SCIM is not explicitly aware of how they should relate to the background distribution. These embeddings seem to be well behaved. The decoder can correctly reconstruct their inputs, and the discriminator is fooled since the embeddings are aligned to the underlying distribution. However, after matching is performed, it's revealed that the blue cells were matched to the red cells and the red to the blue. This is because the encoder mapped the red cells to the blue space, the blue cells to the red space, and the decoder reversed this mapping. This problem arises naturally due to symmetries in the latent space. To overcome this problem, we help SCIM orient the latent space by providing the discriminator with categorical labels that inform it about the underlying structure. These labels could be cell type, for example. Discriminator inputs are concatenated with one-hot representations of these labels. In most applications, only a few labels are enough to successfully orient the technologies. These points act as anchors, defining and coordinating the space around them across technologies. The second challenge we address is model selection under adversarial training. This is in general not straightforward. The practitioner is not afforded some value, such as the negative log likelihood, that can be optimized to select the best parameter settings. Often qualitative inspection or domain-specific metrics are employed to measure the performance of an adversarial model. In SCIM, we use a universal divergence estimator to assess the alignment of embeddings between a pair of technologies. This estimates the symmetric KL divergence between two distributions using only samples from those distributions. It provides the user with a measure of how well SCIM align the latent space between a pair of technologies using only a set of their embeddings. This can be used in conjunction with the reconstruction error to help model selection by identifying successful runs at scale. Once the technology invariant latent space is constructed and cells from all datasets are embedded into it, integration proceeds by pairing similar cells across different datasets using their embeddings. This cell-to-cell matching, unlike a generative approach using the decoder networks, constrains matches to the true data distribution. This choice can reduce downstream errors due to artifacts that could be inadvertently introduced by the generative approach. SCIM performs cell-to-cell matching between technologies pairwise. It operates in the low-dimensional latent space and uses a fast bipartite matching algorithm using a min-cost max-flow approach. The data from two technologies, here circles and hexagons, can then be represented as a bipartite graph, where each cell is a node. To scale to large datasets, we constrain the inter-technology edges to the k-nearest neighbors of each cell. These edges are weighted by the Euclidean distance computed using the low-dimensional embeddings. Finally, to account for statistical differences in the distribution of cells between technologies, we introduce a null node to allow for unmatched cells. This node is connected to all cells in the root technology and is given a large cost and infinite capacity, allowing for but penalizing any unmatched cells. Matching is performed in the usual way, by computing the maximum flow through the graph and then minimizing the cost of this flow. We investigate the performance of SCIM on real-world and synthetic datasets. We first test SCIM's ability to integrate substructure across multiple technologies by simulating data with the PROST tool. PROST simulates single-cell datasets by parameterizing a negative binomial distribution with an underlying temporal branching process. This hierarchical structure is commonly found in single-cell datasets. Each generated cell has a branch label, which determines some overarching structure, and a pseudotime label, which provides a temporal orientation within and between these branches. This parameterization is continuous through pseudotime. Each branch has some distinct signature, but these signatures move continuously in pseudotime between the branches. We define a single tree that is used to generate 3 datasets, each with 64,000 cells and 200 features. The parameters of each dataset's negative binomial distribution is generated using different seeds. This means that the 3 datasets have the same underlying structure defined by this tree, yet their feature sets follow different parameterizations and cannot be directly compared. We choose one dataset as a source technology and initialize the latent space by training a VAE. Then we run SCIM to embed and align all technologies in this shared latent space, supervising using the branch label. The integrated latent space is visualized by computing t-SNE projections on the joint set of embeddings. Each column here shows the t-SNE projection from a single technology, while the rows are colored by branch label and pseudotime, respectively. Since t-SNE projections are computed using the embeddings from all technologies, the axes here have the same orientation and scale. These embeddings reflect the structure defined by the tree and are aligned across all 3 technologies. For example, the blue technologies from branch A interpolate into the green technologies from branch C and the orange technologies from branch B. This is preserved across all embedded technologies. The temporal coloring reveals that interpolations between branches is generally continuous in pseudotime. Next, we investigate the quality of cell matches identified by SCIM. Here, a density plot shows the alignment of pseudotime information across matched cells between the source and a target technology. Correctly matched branches are colored with their branch color, and matches with conflicting branch labels are shown in gray. It can be seen that most branch mismatches occur at the branching points in the tree. Here, the labels are ambiguous since the underlying temporal process is continuous. SCIM accurately matches cells, reporting an overall label accuracy of 86%, and preserves their pseudotime, yielding a Pearson's correlation of 0.86. We've also applied SCIM to a melanoma sample profiled with single-cell RNA-seq and CyTOF technologies, yielding a dataset of over 140,000 cells. Here we show a t-SNE projection of the integrated latent space SCIM produced. A random subset of embeddings are shown. We show that SCIM correctly aligns this latent space between technologies when semi-supervised using only 10% of cell labels. It produces matches of high quality, retaining cell type in 93% of matches. In conclusion, we have developed SCIM, a tool to restore lost cell correspondences in multi-omic datasets. SCIM, short for single-cell integration via matching, proceeds in 2 steps. It first constructs a low-dimensional latent representations of cells that are invariant to their technology. Then it matches cells across technologies using a fast bipartite matching scheme on these representations. SCIM does not require feature correspondences between datasets, and it is applicable to any embeddable data type. It scales to datasets with over 100,000 cells, can integrate multiple technologies at once, and addresses challenges in adversarial optimization. We have a manuscript available on the bioRxiv accessible through this link or QR code. I'd like to thank my collaborators, especially my first co-author, Joanna Fitzek, and our supervisors, Gunnar Ratch and Kyung Lemon. Lastly, our lab has used this web app, Spatial Chat, to hold coffee breaks and other social activities during the quarantine. We thought it might lend itself nicely as a way to mimic an in-person poster session. So for those interested, Joanna and I will be hosting an unofficial, experimental, interactive poster session during the coffee break scheduled directly after the Q&A. You may reach it by following this link. Spatial Chat runs in your browser. And with that, thank you for watching and see you at the Q&A and/or the Spatial Chat.